Today, so I will tell you a few stories, essentially, how to integrate this multi-omic data for disease and population health, essentially. I only have one or two slides for brain models, so for whole brain models, so I will not go into detail for this part, and essentially, say, try to show you a few cases in how can we integrate data together, to look into disease or behavior, such kind of thing. Right, so let me start essentially. And as people are sitting here now, say internationally, so we have a few brain projects going on or just finished actually. Say, as we all know, say in EU we have 10 years brain projects, say HBP, and for 10 years, and it finished last 2 years ago now, yeah. And the USA still say we have a huge kind of joint efforts there, to work on brain. In China, they invested 5 billion Chinese yuan for 10 years. It started a few years back and still going on. So with all these brain projects, and we collect a huge data there, so it's a very rich area for brain science. And so we have a microscopic data from genetic side, from DNA, RNA to protein, to proteomics. And then we also have a cellular level data, how say single neuron recording or multi-unit recording for hundreds or thousands or 10,000 of neurons activity simultaneously. And we also have this macroscopic data available, for example, brain image data. Yeah, so I will talk quite a lot about brain image data, yeah, from structure, activity, and connecting DTI, what we call connections data, all kind of data. And we also have a lot of behavior data, say, say your sleep pattern, for example, etc. And then even more larger scale, we have this environment data, and currently we are working very, actively on this, our image, satellite data, for example. So we have all this data from different scale available, and today what I try to show you is maybe kind of a thing, so how can we integrate us from macroscopic data, yeah, and the mesoscopic data and the microscopic data together, and try to look at brain disease in particular and the behavior side. We have a holistic view. Hopefully, I can show you with all this data available from microscopic to macroscopic data available, so we can give you more, say, in comparison with traditional biological data at one single scale, yeah, and so we can have a more holistic view for disease and behavior, and then we can develop a treatment, for example, for different brain disease, hopefully. Yeah. So that's the thing I want to show you today. So, and that's the purpose I want to show, the central topic I want to show you here. In Fudan University, we are one of the university in Shanghai, we are one of the top university in China in general. Yeah. So we have one of the image center there for belonging to our institute. That's the roof of our image center. And what we did is, so we dig 3,000 square meters underground, and we put our machine, the image machine there, and essentially during the time when I set up this image center, and nature did ask me, what do you want to do? And I told them, well, I'm a computational guy, and one of the folks will be using AI to study brain disease. I think people sitting here might be sure my interest there, say try to using AI to study disease, essentially, yeah. And so we have the largest number of image center in Asia and in Shanghai, in our center essentially. Yeah. And so I collected a lot of data actually quite a few years back now. And so this machine we have currently in our image center. So we have a few 3T machine, 11.7T and 7T and 5T and another 3T there. Yeah, yeah. And not only all these machine, we also have a machine to interfere, to treat the people's brain disorders, for example, like TMS, etc. There, and it's really, say, if you guys sitting there have interest to work with us there, and is more than welcome, to visit us or whatever, we can do work on remotely as well. Yeah, yeah, and for all using the machine there, for all the brain disease. And this data we collected, sorry, I did not translate this into English, but I can translate it now. Yeah. So we have totally collected around 20,000 people's data for schizophrenia, depression, degenerative disease, essentially AD, autism, stroke, and normal health university students. So we have also genetics and image data, behavior data, and such gut microbiome. So all this data available there, and again, the data is openly available. So if you have interest, do send us an email, and we are very happy to share the data with you. Yeah. So with all of this data available, in particular in my group, so what we are doing is, in order to understand what's going on for brain, and so we try to compare healthy brain with diseased brain, and so and as a consequence or as a outcome of this comparison, so we can look into many diseases, and what we try to develop is for prevention, diagnosis, or treatment of many brain health, brain diseases. And for long term, what we try to do is we try to develop AI. So what we call brain-inspired AI in particular. And hopefully, we can implement them in hardware as well. So it's long-term efforts, but today I'm more concentrated on this part. Say, tell you a few stories. Say, how can we work out from this multi-scale data for this purpose? Yeah, and do welcome young people. And we have a lot of exchange with British universities, from Cambridge, Oxford, and also quite a few universities in London as well. In particular, King's College London, say IOP. Yeah, we have many active joint projects going on at the moment. Okay, so as I mentioned, we try to use this microscopic, macroscopic, and microscopic to using them look into the brain disease, and also we also using this environment data a lot, as I mentioned, from very microscopic scale to this very huge scale, especially large scale data looking at brain disease, and also not only in the spatial scale, we also look at the temporal scale. Yeah. So at the end of my talk, hopefully I have time to go back to tell you this whole scale, spatial temporal scale, look at depression, addiction, schizophrenia, OCD, dementia, Parkinson, etc. Today I might be concentrating on a few of them, maybe schizophrenia and dementia in particular. Using the data, how to look at it as a disease. Okay, so let me start from some simple cases first. Let me start from microscopic scale data. So, from DNA, RNA to proteomic data, then look at the disease and the behavior side using all this microscopic data in particular. Today our mainly concentration on proteomics. Actually, we do work on all this. Yeah, we do work on proteomics for genomics and transcriptome a lot. For example, that's we published a few weeks back, or last month for proteomics, and that's another paper and reviewing for Nature Math that look at transcriptome data. But today I will mainly concentrate on possibly tell you the story from proteomics because I think it's a very interesting topic and also it's in comparison with the proteomics, for example, it's more closely related to our disease. So I'll tell you a few stories what we did for proteomics. And the data we use for proteomics is, say, as people all know, recently the clinical progress allow us to have a huge data in the proteomics, for example, here, as I listed here. And in particular, I will tell you a few stories for all-link data, say, that's from blood bank data, and that's the data everyone sitting here, we can publicly access. Yeah. And so yes, that's the example I want to show you today, actually in dementia side. So we have the data from our bank data. Yeah, everyone sitting here, we can have it. Yeah, and when we look at the data, say it's a very interesting data set. Of course, at the moment, as far as we know, we have around say 500,000 people, we have this proteomics data available, and the data we have, I think it's as around, yeah, proteomics, we have around 3,000 proteins available, and the data was collected, say, around 15 years ago. Yeah, so the data we collected is at this point, and so at this point, so we have all the disease, say around 200 disease available. So these people they have these people, yes, they have around 200 kind of different disease, and then we can have we also have the data we can do prediction as well for around 400 disease, and so that's the thing, say, yeah, before the data we collect at this point, so we can, all these people have this 200 disease available, so we can work out the diagnosis issue. Yeah, and for the prediction, we have this 400 disease there, so we can do prediction for this 500,000 people. Yeah. For say, if we only concentrate on the proteomics data, yes, we have 3,000 proteins available. Yeah. So that's the thing we start to look at the data, say, do both the diagnosis and predictions job. I skip this part, this slide essentially, the clinical, the technical side, say we essentially apply all the machine learning or bioinformatics techniques to the data. And the first thing when we look at the data, one of the simplest thing we look at the data is, so for these 3,000 proteins there. And we can do, as I mentioned, we can predict the disease, say, 15 years late, yeah, for using these 3,000 proteins essentially. So we use the machine learning approach. We select, for example, here I show you 11 proteins. 11 and 15 years ago, before the onset of the Alzheimer disease, for example, if you look at this protein, yeah, so that's the onset of the disease, Alzheimer disease, and I think you don't need much help here. Say you can pretty much agree with me. We can predict the disease, say 15 years ago, 15. years ago, pretty much we can do a very good job. Once I tell if we can select these 11 proteins from these 3,000 proteins, and if I could put them together, I'm sure you are pretty much convinced, So yes, this 11 protein, we can do a very reasonable job there. Yeah. So we select these 11 proteins, and we can, say using, say, again, machine learning approach, so we can plus a few other data, demo graphic data and cognitive side data. We can, the AUC we can achieve, we can predict dementia 15 years ago with an AUC around 0.91. It's not bad job at all. Yeah. So using 3 proteins, we can do a pretty similar job already. The AUC could be 0.90 actually. Say to predict the dementia 15 years ago. Yeah. So that's the paper we published last year, almost beginning of last year. And so we published this, and then that's a BBC News, say essentially, or what BBC broadcast, they tell. Well, the news is nothing to do with our paper, but it's before us. But essentially, what I want to tell people, say early dementia diagnosis, using blood proteins, we can predict, we can do a pretty good job. And after the publication, while during the publication, is actually people always ask me, what for? If I want to do prediction, yeah, and that's only they increase people's anxiety. If I can predict, say certain people, say, say age, as older people, for example, say 50 years ago, I can tell you, whether you can whether you will develop into dementia or not. Yeah, 15 years ago, and people always ask me, what for? You increase people's anxiety for these guys. Yeah. But so that's what I told them. If we can diagnose it, normally it's too late. And as people now, we have a few drugs available at the moment. So I really hope, the proteomic side can help people. Say we can using, they can use the drug a bit early, and that might be help them. So that's that's one of the reasons why we are doing it. Say, yeah, and. Okay, so yeah, so we after did this, we essentially develop a similar approach for PD for depression and such, and for PD, for example, we have a quite a similar story published there, and also, I'm very happy to say, it's a publication in the neurology have a comment there, maybe time for a shift in neurologist attitude towards, say like a PD, yeah. So we can use the protein, we can diagnose it early, hopefully, at least there. And also there's a science paper published last year. It's common. It's a paper said, it's a revolutionary high throughput proteomics and AI. Essentially combine proteomics and AI. We can do some good jobs there. For example, this the paper I copied from the paper. This approach along with machine learning analysis, basic proteomics and AI, has led to the revolutionary in understanding of the foundation of a disease. And I do hope so. We can, yeah, do some not good job for the disease prediction, for example, here. Okay, so the publication, what we are doing at the moment, the follow-up thing, we are working with chemists at the moment to develop a protein panels. Say we concentrate on one or three of them, for example, so we can reduce the price from a few thousand Chinese yuan, possibly to 100 yuan. So normal people can be used, for the prediction for healthy people in particular. Yeah. So that's one thing we are doing. And the second thing we follow this up is, so we look into the brain, yeah, for Alzheimer disease, PD or depression, etc. So and what happens if we not only for the brain but all the disease, all the kind of disease, for example, heart disease, lung disease, or whatever. So we use this approach, look at the whole body disease. I'm sure, people sitting there might be more interested. Well, not only interested in our brain disease, but also as part of a disease. So we do work hard, an atlas, they say, work hard, the first atlas for proteins and the disease, essentially, And the paper is published this year as a cover paper, actually. And we have also I have a homepage, large language model homepage there, so people can use it. If you have proteomics data, you can simply upload your data, and as a model can tell you, say what's a potential problem, for you. And also we have a large language model integrated with this prediction, and so can tell you, say, how to prevent and diagnosis, or what are all the treatment sides there. Yeah. So if you have interest, you do please look into have a read of our paper. I don't have I will not go into very the detail for the paper, but someone made a movie for us, about our cell paper there, and again, so essentially we look into, say, around 2000 disease, and we develop this atlas of plasma protein, proteomics, and in health and disease for 50,000 people. Yeah, and so the paper is about, and it's anyway. So yeah, so we do, say, extend our research to the whole body. Okay, and so that's for proteomics data. So we not only look at the proteins only. We also recently extend this to metabolics, genomics, and the chemistry side. We integrate all these data together, and very interesting. I can tell you, well, maybe not surprisingly, the proteomics data does the best job, for predicting. So that's the accuracy, or what's the proportion if we want to predict a disease, for example, as a proportion of the data we used to achieve as a predictive model. And I can tell you, always proteomics data does the best job here. Yeah. So it's a really, I think, it's thanks to the biobank, actually. So we have this proteomics data available, and the proteomics data always does the best job for prediction or diagnosis, actually, for all the diseases we look at. Okay, so that's the microscopic data, so we try to integrate all the omics data together to look into the disease. And also we have not only this macroscopic data, we also have currently in neuroscience, at least, people are trying to collect many macroscopic data, or the neuronal data here, say, to look into the brain disease, for example, from chronic pain to depression. I will not go into detail for this one, but I show you that's another thing for, say, using this cellular data from our institute. It's one colleague is in my institute. So what we are trying to do, we want to have this brain spinal cord interface. So that's essentially, say, we have these people, they have a problem for their spinal cord injury here. Yeah. And so the type we face is, can we record the signal from your brain, bypass this part, this injury part? So if we can implement, kind of, chips implemented here, yeah, so the people can start walk again because the injury here, normally people are paralyzed, so they cannot walk anymore. Yeah. So can we record a signal from your brain and bypass this part, implement the chips in your low part of your spinal cord so people can start to walk or whatever, start walking as normal people. So that's one of the things we did. 4 years back, we started working on this job, essentially it's brain-machine interface, yeah, from brain, but directly they implement chips on your spinal cord. And so the hardware we develop ourselves, say, and the software side, so we develop is decoding a signal from the brain, and then we detect the area, you have an injury there, and we implement the chips there for in the spinal cord, and we implement algorithms so that people can help the people, to start working again. And so the outcome actually is this year, say, so we did the first operation last month, essentially. So we select a few patients there, for example, this guy, and we implemented the chips in his brain in the motor area, actually, and then also implement the chips on the spinal cord, and that's the outcome. Okay. Around 10 years after operation, well, sorry, 10 days after operation, you can see, he started working. We are very happy with it, actually. Say, we using this brain spinal cord interface. Yeah. Essentially, the people tell him, move your left leg or move your right leg in Chinese. Yeah, of course, he right legs. Yeah, move it. Yeah, again, essentially, he's much better now. That's only 10 days after operation, so he's much in a much better situation. And we successfully did the second operation already, and also he started work now. So we are pretty happy with the outcome. So that's kind of thing we are doing at a cellular level, and at the macroscopic level data, so all the image data, I can tell you a few stories again. Again, we use a lot. We work on image data a lot during the past few years. So here, in particular, with schizophrenia, for example, I can tell you one example how we use. How we analyze all the image data. For example, in schizophrenia, one of the questions we asked for the past 10 years around is we always want to find out where's the starting point of your disease. For example, schizophrenia or depression or actually AD as well. So we work on this for the past few years, and that's the publication from America. Our colleagues in America, they collect a huge data called Enigma, for example. They have 4,000 schizophrenia patients available, and when they look into the brain, so they found out that there are many in the brain. We can subdivide the brain into many areas, for example, and then they found out that there's quite a few areas in the brain for schizophrenia patients. In comparison with healthy people, this brain area shrink, yeah, become smaller. For example, like hippocampus, where the memory, yeah, located, and they become smaller, and so they show people, yeah, we have this area, we have a problem in this area, but and actually one of our colleagues from King's College London, one of the very leading expert in this area, and he did send me email, and we had a kind of discussion. He told me, maybe it's not sure, it's all the, there's no shrinkage for schizophrenia patients. Okay, so we started looking into this area, and I collected a huge data, was the largest data in the world essentially. So we collected 1,000 schizophrenia patients from China, and another 4,000 from Enigma from America or internationally. So we have all these patients and healthy subjects available the data. And as I mentioned, the question we ask, I'm not only interested in say, compare as a comparison of the healthy people and schizophrenia patients. What we try to ask is, can you trace back, where's the starting point of your disease? Yeah, using neural image here, the structure data essentially from the scanner. Yeah. So the idea mathematically is quite a simple idea. So we have all these people, they have schizophrenia patients. If we have a large enough samples, yeah, so we can always trace back and say, where's the starting point, where's the first areas they start shrink. Yeah, because we have large enough data in mathematics, we call this algorithmic, so we can always trace back, where's the starting point. And if you can trace back the starting point, and as a consequence, you can subtype them as well, because we have a different starting point of the disease. So follow this idea, and we apply this to schizophrenia, depression. Actually, we did a similar job, and Alzheimer disease, for example. Also, and we are very happy to find out that in schizophrenia we have two subtypes. One subtype of the patients is starting from the shrinkage, starting from the language area. This is part of your brain, so what we call the Broca area. And another subtype of the people, they are shrinkage start from hippocampus. Okay, so we can subtype all the schizophrenia into two subtypes, one starting from language area, from Broca area. Gradually, this will spread to all of the brain, and another subtype start from the hippocampus and gradually spread to all of the brain. So these two subtypes, and then we confirm or validated in many data, for example, from Asia, from Western, from first episode from jackknife for patients. We confirm this findings, and the paper actually we did publish first in Nature Mental Health, first use Asian subtype, Asian population, and we generalize this to the Enigma Enigma data, for the 4,000 schizophrenia patients, and so that's one of the leader from Enigma. They discussing our papers here. Say in one of the airport, I was told, they are discussing the paper there. Okay, so that's the data we use for sectional data only, yes, and we try to assess the schizophrenia subtype based on sectional data. And how about can you confirm this with longitudinal data? So we do have a group of data from Netherlands, actually 6 years follow up, and we first confirm this, say using the data, and we look at many other aspects as well. For example, different treatment for this subtype, etc. But I don't go into detail here. And also not only look at the subtype there, we also, as I mentioned, so in our, say the disease spread from one area gradually, as the disease progress to all of the brain. So we start asking, what's the mechanism for this pathologic spreading? So how is there something, what's the mechanism there, spread from one area gradually to whole brain areas, etc. So we do look into more details into some mechanism side. The paper is just accepted actually. So if you have interest, I'm happy to discuss with you afterwards. Yeah, so actually the story goes back many years now. I still, that's in work. One of our colleagues from Tim Crow from Oxford, actually, he came to my office in work. He told me many years back nowadays, he has this theory, or not him actually. That's one of the founding editor for the brain, and he told us many years back, well, more than 100 years back, and say maybe schizophrenia started from the Broca area, from the language area, but can we, using all the data to prove it or disprove it? Yeah. So that's one of the review paper actually we published a few years back, and we aim for, this purpose to try to prove or disprove this claim. And I'm happy to say, yeah, we have more and more evidence nowadays. Yes, language is one of the starting point. Language area is one of the starting point area, for the schizophrenia. And so we also follow this up at the moment. We try to develop RCT for different treatment with one of our colleagues. Yeah. Very interesting. I show you one slide here. That's one of my colleagues in our institute. They just published a paper last year in Lancet. Actually, the first Chinese prescription in 200 years in Lancet history. And what they prove is one of the traditional Chinese medicine, essentially called 中风醒脑 feng, actually that's a drug called, and they prove it, is essentially is a placebo. So the drug has been used in China for more than 20 years, the Chinese medicine, but unfortunately, they using RCT and they prove, it's not, it's safe to use, but it's essentially is a placebo. So and we working together try to follow our results up, to say how can we develop different treatment for schizophrenia, for example, there. Okay. So we have this microscopic, I tell you, different stories in macroscopic, mesoscopic, and microscopic data. And what we try to, of course, we try to integration all these together, yeah, for the prevention or the treatment. And we did this, say, if you have interest, we did publish a paper last year in British Journal of Psychiatry, try to integrate all these neurotransmitter, genetic environment together for the schizophrenia, for example, and it's published already. So if you have interest, again, you're more than welcome to read it yourself there. Yeah. Okay, so not only this special scale side, we also look at the lifetime, the lifespan across lifespan, how the disease develop, for example, and we have, yeah, it's quite an interesting story going on there, and also the special temporal pattern. But because of time, I don't go into detail there again, say for the disease brain. So essentially, we try to integrate all the different scale data to tell you a more holistic story for brain disease. Yeah, but I'm sure people sitting there, maybe you are not only interested in disease side, but you are more interested in healthy brain. So I tell in the next 10 minutes, I'll tell you a few stories on that side. I try to tell you something maybe you will remember for my talk. Okay, so I go into healthy side. In the past few years, we do work out quite a few kind of popular stories, say, and it's, we publish 9 stories, I think, in the conversation, talking about loneliness, your diet, for example, and sleep side, for example, etc. All these popular stories, and actually one of my colleagues, Barbara Sahakin, Professor Barbara Sahakin from Cambridge, they do publish a book in the last 6 months, essentially. So we had a party there, so for the launch of the publication of the book, and actually, we worked together for quite a few years on this side, and work out all these stories. Essentially, it's many of the book, well, majority of the book stories about our joint work and say how to keep your brain health. Yeah. So I'll tell you one or two stories, show you how we did it. For example, that's one of the things we are doing. So the thing we are working on is and we use brain image, for example. So I have a brain image there. We can record our brain image, and then the question we ask is from the brain image alone, can we decode what the video is? Yeah. So essentially, say we have the brain image there, and we I think I show you more stage 3. Okay. So that's the thing. We have a simultaneous. We have your brain image there, and that's a video you watched using the AI technology at the moment. Can we decode the what's in your mind essentially? Yeah. And I can happily tell you, that's the start of the state of the art work essentially. We published this a bunch of paper last year in AI conference. So using the brain images here, we can decode the video. Will never be 100% true, yes, but semantically, I'm pretty happy to show you and say, okay, to the end already. But I'm very happy to tell you, we can read out as kind of mind reading, say, there's a few papers there, say to read out your brain, what's in your mind. And not only for that, we also develop quite a few techniques for nonlinear association and causality analysis. For example, that's another example I show you. So we collect around 200,000 subjects data, and the x-axis is about your sleep duration. So that's what's meaning here. That's for 10 years old. Sleep less than 7 hours, sleep 8 hours, 9 hours, sleep more than 10 hours, for example, and 11 years old at session or the lifespan. And the y-axis is your depression score, or how well your brain developed at such. We did look at many of them. Yeah. Here, what I show is only depression score. Of course, the smaller the depression score, the better is your brain in a sense here. And if you look at this figure here, and you are say, the figure is monotone. Essentially, the meaning is once more you sleep, the better for your brain. Yeah. So for 10 years old children, 11 years old children, the more you sleep, the better for your brain. That's published a few years back now. Yeah, and when I give a talk on this in China, around 500 million people view it as a video. I'm very happy there, it's for public health, yes, and I say essentially, tell you is the more you sleep, the better is your brain. How about us? I think people sitting here, listen to my talk is more in this range here, and so we did have a look for all these lives, all the sleep duration. In particular, well, if you look at here, for example, around 40 years old, yeah, and very interesting, this curve started bending over, yeah, it become a U shape. So it's kind of, yeah, and if essentially meaning, you sleep too little is not good for your brain. Too much is not good for your brain. The optimal sleeping duration, 7 hours. It's almost universal. Yeah. All of this is saying, 7 hours is the best. This is the optimal sleep duration for your brain. And so we did work on biology data a lot. And with all of this data available, what we try to develop at this moment, also we developed the world's first digital twin brain. We try to simulate our brain with, the brain has 86 billion neurons and has 100 trillion parameters to estimate. So we did publish the paper at the moment, the digital twin brain, and we do hope, to use digital twin brain to for medical application, for example, Alzheimer disease or or this brain machine interface, etc. So that's the thing we developed very recently. And yes, so that's a summary of my talk. What we try to do, we try to compare, try to understand what's going on in our brain, use all the data available to us, and for the prevention, diagnosis, and treatment, etc. And also not only for brain disease, but also for healthy people, and how we can keep our brain healthy. So this side, and yeah, so I think that's my final slide. Yeah, thanks a lot.